Hi everyone, my name is Jordan Pilch and I'm a junior at the University of Arizona, and I'm here to talk to you about Volvox and multicellularity. I'm kind of going to be going through this demonstration with a bunch of pauses in between, but I've added some slides to tell you when you should go through and pause, and I'll wait like 1 or 2 seconds for you to go through and do this, whether or not you're doing this in a classroom or by yourself, just to go through and do it, so that way it's not wasting too much time in case you're doing that transition. But without further ado, let's go ahead and get started. So something about this is that Volvox are cells, as you can tell, because they're a living organism. But before we can even get to that, we have to talk about multicellularity and a little bit about evolution. So why don't you take a second or two and talk to one another about what you know of evolution, or go ahead and write down 1 or 2 sentences about what you know of evolution. So evolution, in the case that we're going to be going through and talking about it today, is the adaptation of organisms to fit within their niche. And I want to say this right off the bat. There is no organism that is more quote unquote complex than any other organism. Some organisms may have single cells, some organisms may have multiple cells, but there is no difference. If they are alive today, they are all as equally adapted and evolved as everything else. And they come to be this way through different pressures like natural selection or artificial selection, like what humans have done. But what we're going to be focusing on today is the transition from multi to uni, or from unicellular to multicellular, my bad. So in addition to talking about what the difference is between those things, why don't you take a minute to go through and talk about what cells are, specifically what some of the differences may be between plant and animal cells. So the important differences that I want to focus on are chloroplasts, which are a organelle which allows the plants to go through and photosynthesize. So they can form sugar and, in this case, oxygen, which they don't need to survive, but they do need the sugar to survive. Animal cells, on the other hand, only have mitochondria to go through and produce energy for them. And they, both chloroplasts and mitochondria, uh, kind of happened to enter into eukaryotic cells by a ancestor of all of the different organisms that have them eating either a chloroplast or a mitochondria ancestor and going, hey, why don't we work together? And now you might think, oh, is that multicellularity? Is that what it is? But that's not quite it. So unicellular organisms are a single cell. And in fact, most life on Earth is just a single cell. The real important difference is that in multicellular organisms, there's cell specialization. This is how you can have the same DNA in all of your cells, but your eyes don't look anything like your hands or your pancreas or your feet. Your DNA is going through and being manipulated in such a way so that it has all the information, but only specific parts of it are being read at the same time. Versus in a unicellular organism, it isn't really dependent upon where in the body it's supposed to be and how specialized it is. It's just, is there food nearby? Is there danger? Let's go through and take care of those situations. But something that we're going to be discussing today is, why did that happen? Why did multicellular life occur? Was it just an increase in diversity? Was it something that allowed them to maybe care for one another better? Did it make it easier to live? We're going to go through and look at some organisms today. And they are all types of algae, and 2 of them are from the genus Volvox, but the other ones are E. elegans and gonium. And they are all, like I said, algae, so they are plant cells, but they're kind of like they're interesting, as these algae can go through and live by themselves. So while they are technically plants, they're kind of like bacterial plants. That's not a scientific way of putting it, but it's a really good conceptual way of seeing it. And you'll kind of see what I'm talking about when I go and show you some videos that I took later on. But they are able to go through and photosynthesize. And I want you to notice something relating to multi and unicellularity as we go through the videos later. And the last thing that I want to talk about is related to that, which is altruism versus symbiosis. So some scientists argue that there is no difference. For definitions, altruism is doing something nice for someone or taking care of someone with no expectancy of benefit in return. It's like hundreds of thousands of years ago. I'm going to go with thousands of years ago. Humans would take care of their sick and their elderly, and they didn't necessarily have a reason to do so. It hindered them. It made it so that way they had to spend a little bit more time taking care of them. And symbiosis is a relationship between two organisms where both organisms benefit. You can think of the birds that sit on the back of rhinos who will alert the rhino to danger, and the birds keep the parasites off of the rhino. So they're also going through and getting the benefit of not having parasites, and the birds get a free meal. So that's kind of symbiosis. And what some scientists argue is that there's no difference between the two of them. And I kind of want you to think about that as we go through this and decide at the end whether or not you think multicellular organisms are symbiotic relationships or altruistic relationships, because I think it's a cool distinction to make between them. So if we were in person, I would be going through and doing this for all of you, but since we aren't, I've gone through and made a little material slide in case your teacher is able to go through and see you in person and wants to do it there. You will need 4 well slides, which look like this, and little cover slips. They're just little glass squares that cover it so that way your microscope doesn't get dirty. You will need a microscope that'll need to go down to at least 40 times magnification. But if you can get down to 100 times magnification, that would be really cool as well. My videos, I can only take at 40 because we had slightly different well covers, so there's a little bit of a difference. And then you will need your algae. We are using the Aruis, which is actually what is pictured here. This is a very nice photo of it. You'll get to see some videos I took later on. The Barbari, E. elegans, and Gonium. That's just a species. You can get whichever kind you want. I don't remember the exact kind that I got because Carolina just said Gonium, and I said, that's fine. This is great. But if you do use them, try and use them rather quickly, just so that way they don't accidentally die, and make sure that they're kept with light because they are photosynthetic. You will also need sterile single-use pipettes, 4 of them because you have 4 different cultures, and the algae really don't do well if they get infected with bacteria, so the sterile nature is really important. This would be the procedure that you would do, or I would do if I was there with you. You would take a well slide, you would take a culture, you would take a cover slip and a pipette. You would take a small sample of the algae culture and you put it in the well part of the slide. You'd cover it with a cover slip, place it on the microscope, and use the coarse and fine, the large and small, focusing knobs to focus it at 10x and then move up to 40x. And then if you want to, you can go up to 100x. And you would just do this with all of the different cultures. And this is what I did to get the videos that I'm now going to go through and show you. And I want you to go through and take some time to like write down what observations you have. This is something that scientists will always go through and do. You will notice that, as Adam Savage said from Mythbusters, it's only science if you write it down. Otherwise, it's just fun. So go through and write down some observations about these, and I'll have little pauses for you to do that as well. So this is Gonium. This is a little video. I want you to go through and notice things about it first before I start talking about it. So I'll let the video play through and then I'll give you a bit of time to write about it. So here's what I notice about Gonium. You can see that they can exist on their own as single cells like this one is, but they can also form single layer clumps with one another. This was taken at 40 times magnification, so this is a very small algae, as you can tell. They're kind of green, but the camera that I was using had its color balance slightly different, so none of them are going to look green necessarily. But they were all taken with a light microscope, so they should be easily available to go through and take. But I want you to keep in mind kind of the being able to live as a single cell and as a larger group of cells as we continue to move on. So this is E. elegans. This video is much shorter, so I'll play it twice for you so that way you can go through and notice it. And once again, just write down some observations that you have. What I noticed about E. elegans is that they are much more in groups. They are forming little clumps, and they're not necessarily as tightly packed as gonium were, and they can still live on their own, as you can see with this cell and this cell, but they are in much larger groups. And that is an important distinction as we now move into the next culture, which I will tell you a little bit more about after you've watched the video. I kind of want you to just see how cool they look, and then I'll tell you some more information. So with V barbari, which is the one that we just looked at, I didn't take this at 40 times magnification. I took this at 10 times magnification. That is 30 times higher. And I was like, okay, these are a little bit bigger than the other ones. These are multicellular. Unlike elegans and gonium, these are multicellular organisms and form these little colonies where there are cells on the inside, as you can kind of see with this one, but there's also a lot of cells on the outside going through and using surface area to their advantage to go through and try and get as much light as possible for the colony to go through and feed it and support it. And that's kind of a really cool thing. And the cells on the outside are also specialized for cell movement as well. So that way they can do all those fast movements that we saw in the video, which is different than the cilia that are on the gonium and elegans, which allowed them to move around. Or more specifically, the gonium. The elegans didn't really want to move around. Now I'm going to show you the last one, and it's personally my favorite just because of how cool it looks. And I'll kind of go into part of the science of why it's cool as well. But once again, just watch the video, tell me what you think, write it down, do whatever your teacher says. It'll be great. I'll give you some time when I pause it later on. So this is Via Ruiz, and I apologize again if I'm pronouncing any of them incorrectly. My second language is Spanish. It is not Latin, so we're trying here. One thing that I noticed about these is that they have these even smaller dark spots inside of them. And it was really nice of this cell to stay perfectly centered there as I take you through this. Those are even more specialized cells. Those are reproductive cells. They're the cells that are going to go off and form more Via Ruiz colonies later on, and they're little groups, and they will be seeded by other colonies as well as they go through and reproduce with one another. You can think of them kind of like the mammalian ovaries and testes, but that's not necessarily the case. It's more like the ovary and angiosperm of a plant, but those are really big words that I barely remember the names of. So if you need a more close analogy, you can use testes and ovaries. But this has even more cell specialization than V. barbari did. And it kind of shows from all the way from gonium to here that there's been a kind of change in how it's looked like. I am going to mention something here. These cultures are all alive today. These are not ancestors of one another. The only ones that are in the same genus are the last two. This is just being used as a model or an approximation of what this process would look like, as they are all algae, so they all have a similar-ish evolutionary background. So we can think of gonium acting in the same way that an ancestor of Via Ruiz might have, but the two are both alive today and they are not ancestors of one another, but they are good models for showing how this works. So now I suggest getting into a breakout room if you're doing this in class and breaking out into about groups of 3 and drawing a model for what you think is happening. And I don't necessarily want you to just draw, here's gonium, here's elegans, here's barbari, here's a Ruiz. I want you to go through and draw arrows on why they did these things or how they came together to form the next step. So maybe you draw 2 gonium coming together, or maybe you draw the way that you think Via Ruiz is going to go through and change later on as well. Be creative and think outside of the box. This is you working through the information that we just got right here. And once you're done, I really want you to go through and share it with other people. If you're working in the classroom, share it with one another. If you're not, you can go through and send it to your teacher, or you can send it to the MCB department here at the U of A. I'm sure that they would absolutely love to see some student work and what you're doing. But go through and share it with someone at least, because it's really important in science to be collaborative. So now that you've discussed it with one another and kind of seen what's going on, I want to take you through what's happening. Part of it is, like I said, moving from unicellular to multicellular. And we know that this happened a long, long time ago because we have evidence of it in the fossil record when photosynthetic organisms started to produce oxygen. They started producing oxygen, and we suddenly had a mass death of most of the life on the planet because oxygen is actually quite toxic, despite basically all life now needing it in some way or another. Um, and after that, we start seeing more multicellular creatures. We see them in the form of like trilobites. We see them in the forms of dinosaurs, but those are much later on than trilobites. This is like very early on in the evolution of multicellular organisms. It's really cool. But I'm also going to let you in on another secret in that I made you do an experiment or look at a demonstration and make observations on something that there is no conclusive answer to. We do not know what's going on. We don't know what's happening, but we do have some hypotheses. I said theories, but that's not quite the right scientific term. But some of the hypotheses that they have are that altruism or symbiosis, whichever one you want to go through and call it, means that they can spend less energy focusing on protecting themselves or gathering food. So by working together, they can go through and limit the amount of energy each one has to spend on reproducing, on finding food, on protecting themselves. Because finding food is, of course, pretty difficult. And if you can have somebody having your back in that situation to give you food, great. That's an even better thing. And sometimes they'll go through and make what are called secondary metabolites, which you can think of them more as like poisons that aren't good for the bacteria to go through and eat, or aren't good for the predators to go through and eat. My apologies. So that's kind of what we're doing, and you should feel very proud of yourself for doing something that scientists elsewhere haven't done either, so it's pretty cool. And with that, I'd like to say thank you for watching. I really hope that you enjoyed my presentation. You can find more from my classmates on probably a page close to mine. And if you have any questions or you want to learn more, please go to the University of Arizona MCB department website and talk with some of the professors, talk with some of the advisors. And I hope that you all have a good rest of your day.